Welcome to Nexus Clinical. Before diving into this lecture, I want to ensure we're all on the same page. This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this content is at the user's risk. This is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay seeking medical advice for any medical condition they have, and they should consult their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures. This content offers a detailed analysis of the regulatory framework established by the U.S. Food and Drug Administration for biological products. It specifically examines the different approval pathways available for innovative biologic drugs, biosimilars, and interchangeable versions of these products. The information is structured to provide clarity on how each category is evaluated and approved within the regulatory system. Biological products have transformed the treatment of many diseases. Unlike small molecule drugs, biologics are complex medicines that are derived from living systems. This fundamental difference requires a special and sophisticated regulatory framework to ensure their safety, purity, and effectiveness. A key concept in this regulation is inherent variation, which refers to the slight and naturally occurring differences that can exist even within a single product from the same manufacturer over time. Because of this, the regulatory approach must shift from demanding strict identity, as is required for generic drugs, to requiring a standard of controlled and acceptable similarity. This analysis discusses the two parallel approval routes established by the Biologics Price Competition and Innovation Act. The first is the traditional pathway for novel reference products, while the second is an abbreviated pathway for biosimilars. The Food and Drug Administration evaluates biosimilar applications using a totality of the evidence approach. This process begins with a highly sensitive comparative analytical assessment, which is then supplemented by targeted clinical data to address any remaining uncertainties. The discussion also highlights the more stringent standard required to achieve interchangeability, which allows pharmacy-level substitution of biosimilars. Furthermore, the analysis examines the economic factors driving the development of biosimilars. It explains the conventions used for product identification, such as the unique four-letter suffix assigned to biosimilars. Finally, the role of clinicians and patient advocacy groups is emphasized, as their support is vital for building confidence and ensuring successful market adoption of biosimilar medicines. To reach this level of understanding, by the end of this module, you will be able to define and differentiate the three distinct categories of biological products within the Food and Drug Administration's regulatory landscape: reference product, biosimilar product, and interchangeable biosimilar product. You will also be able to explain the concept of inherent variation in biologics and how this scientific reality necessitates a regulatory standard of controlled and acceptable similarity rather than strict identity. Additionally, you will distinguish between the purposes and data requirements of the pathway for innovator biologics and the abbreviated pathway for biosimilars. You will learn about the totality of the evidence principle, which provides a flexible, risk-based framework for evaluating biosimilar applications. Furthermore, you will identify the comparative analytical assessment as the foundational and most sensitive part of a biosimilar development program, crucial for detecting potential differences. You will also articulate the fundamental difference in the purpose of clinical trials for biosimilars, which aim to confirm that there are no clinically meaningful differences, versus those for innovative products, which establish safety and efficacy from the outset. The module will cover the additional stringent statutory requirements that a product must meet to be designated as an interchangeable biosimilar, as well as the importance of a dedicated switching study in providing confidence for pharmacy-level substitution of such products. You will understand the unique non-proprietary naming system for biologics, which includes a four-letter suffix to ensure unambiguous product identification and enhance pharmacovigilance. Lastly, you will recognize the Food and Drug Administration's Purple Book as the definitive database for verifying a biologics license type and its interchangeability status. The emergence of biological products has transformed the treatment options for a variety of diseases, including chronic inflammatory conditions, cancers, and metabolic disorders. Unlike traditional small molecule drugs, which are usually produced through chemical synthesis, biologics are a diverse and complex category of medicines derived from living organisms. This fundamental difference in origin and structure requires an equally sophisticated regulatory framework to ensure their safety, purity, and effectiveness. In the United States, the Food and Drug Administration has established comprehensive pathways and standards that recognize the unique scientific characteristics of biologics. These regulations oversee the approval process for original biologic medicines, as well as their follow-on versions known as biosimilars and interchangeable products. To fully understand this regulatory framework, it is important to start with an appreciation of the intrinsic nature of biologics themselves. The nature of biologics involves the complexity and inherent variation found in these medical products. Biological products, commonly known as biologics, encompass a wide range of medicines including therapeutic proteins, growth factors, hormones, and monoclonal antibodies. These products are generally large and complex molecules that are produced within living systems such as microorganisms, which include bacteria and yeast or animal cells. The production process often utilizes recombinant DNA technology. This manufacturing process is inherently more complex than the chemical synthesis used for small molecule drugs, making the resulting biologic products more difficult to fully characterize and purify. A defining characteristic of all biologic products is the presence of inherent variation. This term describes the subtle, naturally occurring differences that exist within a single manufacturing batch as well as between different batches of the same product. Such variation is an unavoidable result of producing these substances in living cells. For example, in the manufacturing of therapeutic proteins such as monoclonal antibodies, cells introduce minor changes to the proteins after they are synthesized. This process is known as post-translational modification. These modifications can include the attachment of biochemical groups, such as sugar chains, or other alterations like the removal of amide groups, the formation of alternative disulfide bonds, or truncation of terminal amino acids. These variations can also result from specific manufacturing procedures or storage conditions. The concept of inherent variation is the most critical scientific principle underpinning the entire regulatory framework for follow-on biologics. This concept requires a fundamental shift away from the traditional standard of identity used for small molecule generic drugs. Generic drugs are chemically synthesized and have active ingredients that are identical across different production lots. As a result, the regulatory approval process for these drugs is based on demonstrating bioequivalence. In contrast, biologics, even those produced by the original innovator, exhibit natural lot-to-lot variability. It is scientifically impossible to demand that a follow-on biologic be completely identical to a reference product that itself is not even identical over time. This reality necessitates an evolution in the regulatory paradigm, moving from a focus on sameness to one of controlled and acceptable similarity. The regulatory challenge is no longer to eliminate variability, but to define, measure, and compare the patterns of variation. This involves ensuring that the variations between a proposed biosimilar and its reference product fall within a scientifically acceptable range. Such an approach allows for a more realistic and rigorous assessment of biosimilarity, acknowledging the natural variability inherent in biologic products. Within the U.S. Food and Drug Administration's regulatory landscape, biologic products are categorized into three distinct groups, each with specific definitions and roles in clinical practice. One of these categories is the reference product, which refers to the original biological product that the Food and Drug Administration has approved through a comprehensive, standalone biologics license application. This product serves as the benchmark against which proposed biosimilar or interchangeable products are evaluated. The approval of a reference product is based on a complete package of data demonstrating its safety and effectiveness for its approved uses. Examples of reference products include Humira, a monoclonal antibody used to treat various autoimmune conditions such as rheumatoid arthritis, psoriasis, and Crohn's disease. Remicade, another monoclonal antibody, is employed to treat autoimmune disorders like Crohn's disease, ulcerative colitis, and rheumatoid arthritis. Lantus is a long-acting insulin analog used to manage diabetes. Herceptin is a targeted therapy for certain types of breast and stomach cancer. Avastin, a monoclonal antibody, is used to treat various cancers, including colorectal, lung, and kidney cancers. A biosimilar is a biological product that is demonstrated to be highly similar to a Food and Drug Administration-approved reference product, despite minor differences in clinically inactive components. Moreover, a biosimilar must show that there are no clinically meaningful differences in terms of safety, purity, and potency, which includes safety and effectiveness, compared to the reference product. Like the original product, a biosimilar is prescribed by a healthcare provider. All biosimilars are subject to the same rigorous approval standards by the Food and Drug Administration as their reference products, ensuring that patients and healthcare providers can have confidence in their use. Examples of biosimilar products include Inflectra, which is a biosimilar to the reference product Remicade, Vasai, which is similar to Avastin, Zarxio, a biosimilar to Neupogen used to increase white blood cell counts, and Amjevita, which is comparable to Humira. An interchangeable biosimilar product is a biosimilar that has met additional, more stringent legal requirements. In addition to demonstrating biosimilarity, the manufacturer must provide sufficient data to prove that the product is expected to produce the same clinical result as the reference product in any given patient. For products that are administered more than once, the data must also show that the risk, in terms of safety or reduced effectiveness, associated with alternating or switching between the interchangeable product and the reference product is not greater than the risk of using the reference product without such a switch. A significant practical distinction of an interchangeable product is that it may be substituted for the reference product at the pharmacy level by a pharmacist without the direct intervention of the prescribing healthcare provider, provided that it complies with individual state pharmacy laws. Examples of interchangeable biosimilar products include Semglee, which is insulin glargine YFGN, and Resvigler, also insulin glargine Aglar. Both of these are interchangeable biosimilars to the reference product Lantus. Siltizo, an interchangeable biosimilar to Humira, contains adalimumab. Cimerli, which is an interchangeable biosimilar to Leqembi, is used to treat certain eye conditions and contains ranibizumab. It is important to note that all three categories of biologic medications, reference products, biosimilars, and interchangeable biosimilars, are considered safe and effective by the Food and Drug Administration for their approved uses. The introduction of biosimilars and interchangeable biosimilars has the potential to increase patient access to essential biologic therapies. This can be achieved by fostering competition within the healthcare market, which may also lead to a reduction in costs associated with these treatments. The Biologics Price Competition and Innovation Act of 2009 established a legal framework in the United States for the approval of biosimilar and interchangeable biological products. This legislation introduced an abbreviated pathway for approval, which is detailed in Section 351(k) of the Public Health Service Act. This pathway exists alongside the traditional, standalone approval process for innovative biological products, found in Section 351(a) of the same Act. These two approval processes are founded on fundamentally different philosophies and require different types of evidence to support the approval of a biological product. The pathway described under Section 351(a) involves the approval process for innovative biological products. When a manufacturer seeks approval through this pathway, they must submit a standalone biologics license application. This application must provide a comprehensive and independent demonstration of the product's safety and effectiveness for each of its proposed uses. Once a biological product is approved via this pathway, it becomes the reference standard for future biosimilars, setting the benchmark for subsequent developments in the field. The data package for a 351 application is comprehensive and designed to establish a thorough scientific understanding of the product from the very beginning. It generally includes extensive data from four main areas. The first area is product quality, which encompasses detailed chemistry, manufacturing, and controls data that characterize the molecule and the manufacturing process. The second area involves animal and non-clinical studies, consisting of preclinical data obtained from laboratory and animal tests to evaluate pharmacology and toxicology. The third area focuses on clinical pharmacology, which includes studies conducted in humans to understand how the body affects the drug, known as pharmacokinetics, and how the drug affects the body, known as pharmacodynamics. The final area comprises safety and efficacy studies from large-scale, rigorous clinical trials. These trials are often randomized and controlled, conducted within relevant patient populations to demonstrate that the product is safe and effective for each specific disease or condition for which approval is being sought. The significant investment of time and resources required for a biologics license application under Section 351A serves a purpose beyond the approval of a single product. This rigorous process establishes the definitive scientific and clinical benchmark for that particular biologic. The data generated through this pathway, characterizing the molecule's structure and function, defining its clinical profile in patients, and identifying its risks and benefits, creates the foundational knowledge base that makes the abbreviated pathway for biosimilars possible. The biosimilar pathway is based on the ability to leverage the Food and Drug Administration's prior finding of safety and effectiveness for the reference product. This finding could only have been established through the exhaustive data package of a biologics license application. In this sense, the biologics license application pathway does not simply approve a drug. It establishes a public, scientifically validated standard of care. The 3-5-1-K pathway was specifically established to offer a shortened approval process for biological products that are proven to be similar or interchangeable with an United States Food and Drug Administration licensed reference product. The main aim of this pathway is to enhance patient access to biological therapies by fostering competition, which can result in more therapeutic options and lower costs. It accomplishes this by decreasing the time and expenses involved in development, mainly by avoiding the necessity to duplicate the extensive and costly clinical effectiveness trials already conducted for the reference product. The primary goal of a development program following this pathway is not to independently verify the safety and effectiveness of the proposed product. Instead, it is to demonstrate biosimilarity through a comprehensive and direct comparison to the reference product. Consequently, the data package is inherently comparative. It is based on a foundation of detailed analytical studies, followed by targeted clinical pharmacology studies, and if needed, additional clinical studies to address any remaining questions. When a proposed product successfully meets the high standards for biosimilarity under this pathway, it is understood to carry the same risks and benefits as its reference product for the approved conditions of use. The following table provides a clear, side-by-side comparison of the key distinctions between the 3-5-1-A and 3-5-1-K regulatory pathways. The 3-5-1-A pathway and the 3-5-1-K pathway are two distinct regulatory pathways for biological products. The 3-5-1-A pathway is for a standalone biologics license application, BLA, and is used for reference products, innovator biologics. Its primary goal is to independently establish the safety and effectiveness of a novel product for each proposed indication. The foundational evidence for this pathway includes product quality data, supported by extensive non-clinical and clinical data packages. A full complement of clinical safety and efficacy trials is required to support each indication being sought. The approval outcome is an independent FDA finding of safety and effectiveness for the product. In contrast, the 3-5-1-K pathway is for an abbreviated biologics license application, BLA, and is used for biosimilar or interchangeable products. The primary goal of this pathway is to demonstrate that the proposed product is highly similar to and has no clinically meaningful differences from an approved reference product. The foundational evidence relies on a comprehensive comparative analytical assessment, that directly compares the proposed product to the reference product. Clinical data requirements include targeted clinical studies designed to address any residual uncertainty about biosimilarity after the comparative analytical assessment, which may not require large efficacy trials. The approval outcome leverages the FDA's previous determination of safety and effectiveness for the reference product, with the approval confirming biosimilarity. The Food and Drug Administration evaluates a biosimilar application based on the principle of considering the complete set of evidence. This means that the agency does not rely on any single piece of information. Instead, it performs a comprehensive assessment of all available data to determine whether the biosimilarity standard has been achieved. The evidence reviewed includes product quality assessments, non-clinical data, clinical pharmacology studies, immunogenicity evaluations, and any other clinical studies that may be available. This principle establishes a flexible and risk-based framework for the development of biosimilar products, rather than following a rigid, one-size-fits-all checklist. The development process is designed as a stepwise progression, where the amount and type of evidence needed in the later stages are directly influenced by the strength and certainty of the evidence obtained in earlier stages. The process begins with the most sensitive and foundational type of evidence, known as the comparative analytical assessment. This step involves a thorough comparison of the analytical and functional characteristics of the biosimilar candidate with the reference product. Subsequent clinical studies are then specifically tailored to address any remaining uncertainties. These uncertainties may persist after the comprehensive analytical and functional characterization are complete, and the clinical studies aim to resolve them effectively. This approach has significant practical implications for drug development. When the comprehensive analytical assessment is exceptionally thorough and shows a high level of structural and functional similarity between the proposed biosimilar and the reference product, the remaining uncertainties are typically minimal. As a result, the subsequent clinical studies needed to resolve these uncertainties can be more focused and less extensive. On the other hand, if the analytical comparison uncovers subtle differences whose clinical significance is unclear, a more extensive set of clinical data may be necessary to prove that these differences do not impact the safety or efficacy of the product. The holistic approach to evaluating evidence enables the Food and Drug Administration to consider the entire data package. This allows for tailoring the requirements to address the specific scientific questions related to each individual product, creating a regulatory pathway that is both efficient and rigorous. Demonstrating the similarity between biosimilar products is a rigorous scientific process that advances step by step. It begins with a thorough molecular characterization of the products involved. This initial phase is crucial for understanding their fundamental properties. Following this, confirmatory clinical data are collected to support the comparability. The evidence supporting biosimilarity is structured like a pyramid, with the most sensitive analytical methods forming the foundation. This approach allows for a focused and efficient resolution of any remaining questions regarding the comparability of the two products. The comparative analytical assessment serves as the fundamental basis for every biosimilar development program. The U.S. Food and Drug Administration considers this the most sensitive part of the evaluation process for identifying potential differences between a proposed biosimilar and its reference product. This assessment is often capable of detecting subtle variations that might be imperceptible in human clinical trials. The scientific principle underpinning this approach is that a product which is demonstrated to be highly similar in structure and function to its reference product through comprehensive analytical testing is expected to behave similarly in a clinical setting. The assessment involves an extensive series of advanced analytical techniques that facilitate a direct, side-by-side comparison of the proposed biosimilar with multiple batches of the reference product. This comparison is thorough, covering a broad spectrum of quality attributes. These attributes include physical, chemical, or biological properties that could influence the product's clinical performance. The selection of attributes for evaluation is based on their known or potential impact on the biological activity of the product, as well as its pharmacokinetic and pharmacodynamic profiles. Safety, efficacy, and immunogenicity are also considered crucial factors in this comprehensive analysis. The reliance on the comparative analytical assessment as the cornerstone of biosimilar approval is a direct reflection of decades of advancement in analytical biotechnology. Modern techniques such as high-resolution mass spectrometry, various forms of chromatography, and sophisticated bioassays are now so powerful that they can provide an incredibly detailed picture of a biologic's molecular structure and function. This bottom-up approach, starting with a fundamental comparison of the molecules themselves, is more scientifically rigorous and efficient than a traditional top-down approach that begins with clinical outcomes. By establishing a high degree of analytical similarity first, the subsequent clinical studies can be designed to be confirmatory rather than exploratory, focusing on targeted questions to ensure the absence of clinically meaningful differences. For a complex protein such as insulin, a comparative analytical assessment would involve evaluating numerous quality attributes across multiple domains. One important aspect is the molecular structure, which includes confirming the primary structure by analyzing the amino acid sequence and molecular mass. Additionally, it involves assessing the higher-order structure, such as the protein's conformation and the presence of disulfide bonds, as well as characterizing any post-translational modifications. Another important domain is the identification and quantification of product variants. These can include forms of the protein that have undergone modifications such as deamidation or acetylation. Bioactivity is also a critical attribute, assessed through various functional assays. These assays compare biological activity by analyzing receptor binding kinetics and downstream cellular effects, such as glucose uptake in the case of insulin. Lastly, the assessment includes measuring the purity of the protein, which involves determining the protein concentration and quantifying impurities related to the product. Such impurities may include aggregates or high molecular weight proteins that could affect the safety and efficacy of the product. To improve confidence in the assessment process, manufacturers of biosimilar products are highly encouraged to utilize orthogonal methods. These are independent analytical techniques that measure the same attribute of the product but do so in different ways. Using such methods as a best practice helps ensure the accuracy and reliability of the assessment. Following the foundational comparative analytical assessment, clinical studies are conducted to address any remaining uncertainties. These uncertainties are also known as residual uncertainty. The purpose of these studies is to confirm that there are no clinically meaningful differences between the proposed biosimilar and the reference product. This confirmation pertains to both safety and effectiveness. The specific nature, scope, and number of clinical studies required depend directly on the extent of residual uncertainty identified during the preceding analytical and non-clinical evaluations. It is important to understand that the purpose of clinical trials in the development of biosimilar products differs significantly from that in the development of innovative drugs. When developing an innovative drug, clinical trials are primarily designed to establish safety and effectiveness from the beginning. These trials usually involve demonstrating that the drug works for a specific disease, often by comparing it to a placebo or a standard treatment. On the other hand, for biosimilar products, the effectiveness of the original reference product has already been established through previous research. Therefore, the main goal of clinical trials for biosimilars is not to prove that the product is effective, but to confirm that there are no meaningful differences in clinical outcomes when comparing the biosimilar directly with the reference product. This approach involves conducting equivalence or similarity trials, which use statistical methods to demonstrate that the two products are comparable within a predetermined margin. Unlike trials designed to prove superiority or non-inferiority, these studies focus on establishing that the biosimilar and the reference product are essentially alike. This focus allows for smaller and more targeted clinical studies, which can be more efficient in terms of resources and time. Clinical pharmacology studies, pharmacokinetics and pharmacodynamics, are an essential part of developing a biosimilar. These studies compare how the biosimilar and the original product behave in the body and how they affect the body. Pharmacokinetics involves measuring how a drug is absorbed, distributed, metabolized, and eliminated, providing an understanding of the drug's concentration in the body over time. Showing similarity in pharmacokinetics is crucial for demonstrating that the biosimilar will reach its target in the same way as the original product. Pharmacodynamics assesses the drug's physiological effects on the body. If there is a relevant and sensitive biological marker, such as neutrophil count for drugs like filgrastim, it can be used as an indirect measure of clinical effectiveness. These pharmacokinetic and pharmacodynamic measurements are often more sensitive than broad clinical efficacy outcomes in detecting differences between products. When a suitable biological marker is available, manufacturers can use a combined approach that includes pharmacokinetics, pharmacodynamics, and immune response assessments to demonstrate biosimilarity, potentially avoiding the need for large clinical efficacy trials. When a suitable and sensitive biomarker is not available to predict the clinical effectiveness of a pharmaceutical product, it may be necessary to conduct a comparative clinical study in a relevant patient population. This approach, which combines pharmacokinetic analysis with a clinical study, uses the clinical study to provide essential evidence demonstrating comparable effectiveness and safety. The study should be designed with enough sensitivity to detect any potential clinically meaningful differences in effectiveness, safety, and immune response between the biosimilar and the original reference product. Immunogenicity assessment is a vital safety consideration for all biological products. It refers to the potential of a biological product to trigger an immune response in the body, such as the production of anti-drug antibodies. Conducting a comparative immunogenicity assessment is a crucial part of every biosimilar development program. The goal is to ensure that the proposed biosimilar produces the same type and amount of immune response as its reference product. If a patient develops anti-drug antibodies that neutralize the effect of the reference product, it is expected that they would have a similar response to the biosimilar. In such cases, switching to the biosimilar would not be beneficial. Typically, this assessment is integrated into the comparative clinical studies to evaluate immunogenicity. While all biosimilars must meet the FDA's rigorous standards for safety and effectiveness, the interchangeable designation represents a higher tier of approval. This designation has significant practical implications, particularly at the pharmacy level, and requires a manufacturer to meet additional, specific statutory standards supported by further clinical data. The standard for interchangeability set by the Food and Drug Administration requires that a product be licensed as an interchangeable biosimilar if it meets certain stringent criteria. Firstly, the product must be highly similar to the reference biological product, with no clinically meaningful differences. In addition, the manufacturer must provide data to satisfy two further statutory requirements. The first is that the interchangeable product is expected to produce the same clinical result as the reference product for any given patient. The second is that, for biological products that are administered more than once to a patient, the risk associated with switching between the interchangeable product and the reference product in terms of safety or reduced efficacy must not be greater than the risk of using the reference product alone without such switching. A manufacturer must specifically seek this designation in its application. A product is not automatically considered for interchangeability. The role of switching studies is crucial in meeting the second, and arguably most complex, requirement for the interchangeability of biosimilar products. To achieve this, a manufacturer's application must contain additional data and information that specifically evaluate the impact of switching or alternating between the biosimilar and the reference product. This evaluation generally necessitates a dedicated switching study, which is a clinical trial designed to replicate real-world scenarios. In such scenarios, a patient might switch back and forth between the two products multiple times. The idea of interchangeability and the need for switching studies directly respond to current pharmacy practices. Although physicians can prescribe any FDA-approved biosimilar, pharmacists can automatically substitute an interchangeable biosimilar for its reference product without contacting the prescriber, depending on state laws. This means a patient might receive the reference product at one fill and the interchangeable product at the next, and possibly multiple times. An important scientific question arises: is it safe and effective for a patient's treatment to switch between these two similar but not identical biological products? Switching studies are designed to answer this, giving prescribers, pharmacists, and patients confidence that such substitutions won't compromise safety or efficacy. Thus, the interchangeability designation is more than just a mark of analytical similarity. It specifically approves repeated, unmonitored pharmacy-level switching. It is important to note that healthcare providers do not need to wait for a biosimilar to be designated as interchangeable before prescribing it. All approved biosimilars have been shown to be as safe and effective as their reference products. To ensure healthcare professionals can confidently and safely use biosimilars, the Food and Drug Administration has established clear conventions for product identification. Additionally, it provides accessible resources to obtain regulatory information. These tools are essential for accurate prescribing, dispensing, and monitoring of biosimilars after they have entered the market. The non-proprietary naming convention has been established by the Food and Drug Administration to promote safe use and improve pharmacovigilance of all biologic products. This naming system for biologics, including biosimilars and interchangeable products, consists of two parts. The first part is a core name that is common to the reference product and all of its biosimilars. For example, the core name might be filgrastim or replicamab. The second part is a unique, hyphenated, four-letter suffix that has no specific meaning and is distinct for each manufacturer's product. An example of this suffix could be CGT. Here is a revised version of the text with improved sentence structure and paragraph formatting, and with abbreviations removed. For example, a reference product might be named replicamab HGXF, while a biosimilar to it might be named replicamab CGT. This naming system ensures that all related products are clearly linked by a shared core name, while each specific product remains unambiguously identifiable. All biosimilar products and interchangeable biosimilar products are required to have a unique suffix added to their names. To prevent confusion, some older reference products that were approved before the implementation of this naming convention may not have a suffix. This unique four-letter suffix is far more than a simple identifier. It is a critical tool for pharmacovigilance, which is the science and activities relating to the detection, assessment, understanding, and prevention of adverse effects or any other drug-related problem. Given that all biologics have inherent variation and that a biosimilar is manufactured through a different process than its reference product, the ability to accurately track adverse events to a specific product is paramount. If multiple biosimilars for the same reference product are on the market, an adverse event report for replicamab would be ambiguous. The unique suffix, CGT versus HGXF, removes this ambiguity, allowing regulators and manufacturers to precisely attribute any safety signals. This enhances the safety monitoring of the entire class of products and protects public health. The prescribing information, also known as the product label, is the primary tool used by the Food and Drug Administration to communicate essential scientific information to healthcare professionals. This information is crucial for ensuring the safe and effective use of a medication. When it comes to biosimilar or interchangeable products, their labeling is based on and includes relevant data and information from the label of the reference product. This includes the clinical data that supported the original approval of the reference product. However, a biosimilar's label does not have to match that of its reference product exactly. For example, a biosimilar may be approved for fewer indications or dosing regimens than the reference product. Therefore, healthcare professionals must review the specific product labeling to understand a biosimilar's approved uses. A key element in the labeling for all biosimilars and interchangeable products is a specific statement located in the Highlights of Prescribing Information section. This biosimilarity statement, or interchangeability statement, clearly explains the product's relationship to its reference product and outlines the basis for its approval. The prescribing information for a biosimilar does not need to include the detailed comparative analytical and clinical data that the manufacturer submitted to demonstrate biosimilarity. That comprehensive scientific information is publicly available in the FDA's review documents, which can be accessed through the Drugs at FDA database. To provide patients, clinicians, and pharmacists with a single, authoritative source of information on licensed biological products, the FDA maintains the Purple Book. The Purple Book is a user-friendly, online, searchable database that contains information about all FDA-approved biologics, including therapeutic proteins, vaccines, and cellular and gene therapies. Crucially, The Purple Book allows users to identify whether a specific biologic is a reference product, a biosimilar, or an interchangeable biosimilar. It is the definitive resource for determining a product's interchangeability status. The FDA has determined that this information is more appropriately housed in the Purple Book, which serves as a pharmaceutical reference, rather than in the prescriber-focused product labeling. The database provides key details such as the product's proprietary, brand, and proper, non-proprietary names, its biologics license application, BLA, number, and its license type, either 3-5-1-A for a reference product or 3-5-1-K for a biosimilar or interchangeable product. The Purple Book is not merely an informational database. It is a critical piece of the national regulatory infrastructure. Pharmacy practice is regulated at the state level, and many state laws governing automatic pharmacy-level substitution of biologics rely on a clear, official source to define which products are eligible for such substitution. The Purple Book is that single source of truth. By centralizing the official designation of interchangeability, the FDA provides the unambiguous data point necessary for state-level regulations to function consistently and safely, thereby enabling the entire system of interchangeable substitution across the country. The development and approval of biosimilars were driven by a significant economic imperative to introduce competition into the market for biologic medicines, which are among the most expensive pharmaceuticals. Biologics account for a disproportionately high share of pharmaceutical spending in the U.S., representing only 2% of prescriptions but 40% of total spending. The goal of the BPCIA was to create a pathway that would lead to lower prices, reduced healthcare system costs, and increased patient access to these vital therapies. Since the first biosimilar launch in the U.S., the market has generated significant savings. In 2023 alone, biosimilars saved the U.S. healthcare system $12.4 billion, contributing to a total of $36 billion in savings since 2015. Projections for 2024 nearly double the annual savings to $20.2 billion, bringing the overall total to $56.2 billion since market inception. Looking ahead, various analyses project ongoing, substantial savings, with some estimates exceeding $100 billion over 5-year periods. These savings are achieved not only through the lower prices of biosimilars, but also by the downward price pressure they place on the reference biologics themselves. Despite the significant savings potential, the adoption of biosimilars in the U.S. has been slower and more uneven than in Europe. Uptake varies dramatically by molecule. For example, biosimilars for the cancer therapies bevacizumab and trastuzumab have captured over 80% of their respective markets, whereas biosimilars for infliximab and insulin lispro have seen much lower adoption rates. Even within a successful therapeutic area like oncology, uptake is not universal, demonstrating that factors like prescriber comfort and contracting play a major role. Several systemic barriers contribute to the slower-than-expected adoption of biosimilars. One significant obstacle involves practices by pharmacy benefit managers and insurers. These entities can create barriers that favor higher-priced reference products through complex rebate systems, sometimes called rebate walls. Such financial incentives can limit access to lower-cost biosimilars for formulary inclusion. Another challenge stems from patent and legal tactics employed by originator biologic manufacturers. These companies often create dense patent thickets and participate in litigation, strategies that can delay the entry of approved biosimilars into the market. A further issue is the so-called biosimilar void. Over the next decade, 118 biologic drugs are expected to lose their patent exclusivity, representing a potential market value of approximately $234 billion. However, only 12 of these biologics currently have biosimilars in development. This indicates that about 90% face no competition, which threatens future access and potential savings. Addressing these barriers is critical to realizing the full economic and public health promise of biosimilars. The successful integration of biosimilars into clinical practice hinges on the confidence and willingness of healthcare providers to prescribe them. This acceptance is shaped by clinical data, real-world experience, and guidance from leading professional medical societies. Physician attitudes and perceptions towards biosimilars are generally positive. Surveys conducted in the United States indicate a high level of confidence among physicians regarding the fundamental principles of biosimilars. A survey from 2022 revealed that 92% of physicians who prescribe biological medicines were confident in the safety and effectiveness of biosimilars. Additionally, 89% of these physicians expressed willingness to prescribe biosimilars to new patients. This strong confidence among physicians plays a vital role in fostering patient acceptance, as studies have demonstrated that patients tend to trust their healthcare providers' recommendations highly. Despite growing familiarity with biosimilars, significant nuances and concerns remain among physicians. For instance, while about 80% of physicians feel comfortable initiating a switch from a reference biologic to a biosimilar for a patient who is stable, they are much less comfortable with such a switch being initiated by a third party, such as an insurance company, for reasons unrelated to the patient's medical condition. The majority of physicians believe that the decision to switch should be a collaborative effort between the doctor and the patient. In addition to clinical concerns, there are notable knowledge gaps. Many physicians lack detailed understanding of specific aspects of biosimilars, such as what it means for a drug to be interchangeable according to regulatory standards, or the laws at the state level regarding substitution. These uncertainties are especially pronounced in certain medical specialties. For example, surveys indicate that dermatologists tend to be more hesitant to prescribe biosimilars compared to other specialists, often citing concerns about their safety and effectiveness. Furthermore, operational barriers also impact physicians. These include challenges related to navigating insurance coverage policies, ensuring access to support services for patients, and managing the logistical aspects of stocking multiple similar products in their practices. Leading medical organizations have developed position statements and educational resources to guide clinicians, support patient safety, and advocate for policies that promote a sustainable market for biosimilar medicines. The American Medical Association supports the development of biosimilars as a means to increase patient access to these medicines and reduce costs. The organization emphasizes the importance of physician autonomy in treatment decisions and advocates for a clear and predictable regulatory process. Recognizing the advancements in scientific evidence, the American Medical Association has updated its stance to support removing the requirement for switching studies needed to designate biosimilars as interchangeable, a change intended to lower barriers to access. The American Society of Clinical Oncology has been a leader in educating the oncology community. It has issued policy statements affirming that biosimilars and their reference biologic products are equally effective for use in clinical practice. The guidance provided by this organization addresses critical issues such as the interchangeability of biosimilars, policies related to health insurance payers, and the necessity of ongoing education for both healthcare providers and patients to build confidence and overcome obstacles to adoption. The American College of Rheumatology has shifted from a cautious initial position to now recommending that healthcare providers incorporate biosimilars into treatment plans for patients with rheumatic diseases. The organization strongly believes that decisions regarding switching a patient's therapy should be made collaboratively by the prescriber and patient. It opposes non-medical switching dictated by insurers. Along similar lines, the American College of Rheumatology supports the Food and Drug Administration's proposal to eliminate the requirement for switching studies to attain an interchangeability status, viewing this as an unnecessary barrier to patient access. The American Academy of Dermatology and the National Psoriasis Foundation collaborate to develop clinical guidelines for the use of biologic medicines in treating psoriasis. They also provide educational resources aimed at dermatologists and patients. Their efforts emphasize the importance of addressing knowledge gaps among dermatologists to improve the adoption of biosimilars in their field. While regulatory approval, economic factors, and clinical acceptance are essential, the ultimate success of biosimilars depends on the trust and confidence of patients. Understanding patient perspectives and the role of advocacy organizations is crucial for the successful integration of these medicines into the healthcare landscape. Patient awareness and concerns remain a significant challenge despite the increasing availability of biosimilar medications. Many patients are unfamiliar with the term biosimilar or do not fully understand its meaning. For instance, a survey conducted by the Arthritis Foundation revealed that nearly half of the respondents had no familiarity with the term. This lack of awareness highlights the need for better education and communication efforts to inform patients about biosimilars and their potential benefits. The lack of familiarity with biosimilars often raises specific concerns among patients. One major issue is safety and efficacy. Patients frequently worry about whether a biosimilar will be as safe and effective as the reference biologic they are accustomed to, especially if they are stable on their current treatment. Another significant concern involves non-medical switching, which refers to changing a patient's medication due to reasons unrelated to medical necessity, such as insurance policy changes. A survey conducted by the Crohn's and Colitis Foundation revealed that nearly half of patients with inflammatory bowel disease were worried about switching their medication to a biosimilar. Furthermore, patients overwhelmingly want to be informed and involved in any decisions regarding medication changes. A survey indicated that 85% of patients desired to be notified if they were to receive a biosimilar instead of their prescribed biologic medication. Critically, education is key to building confidence. Studies show that while patients may have initial reservations, their willingness to use a biosimilar increases with more information, especially when that information comes from a trusted source. The most trusted source of information for patients is their healthcare provider. Research shows that a physician's recommendation is the single most important factor in a patient's acceptance of a biosimilar. The role of patient advocacy and education is crucial in healthcare. Organizations dedicated to patient advocacy serve an essential function by bridging the knowledge gap for patients and caregivers. For example, groups such as the Arthritis Foundation, the Crohn's and Colitis Foundation, and the National Psoriasis Foundation provide valuable educational resources that are accessible and easy to understand. They also offer support to those affected by various health conditions, helping patients and their families better manage their health and navigate the healthcare system. These organizations serve several important functions. They develop a wide range of educational resources, including websites, fact sheets, electronic books, and videos. These materials explain what biosimilars are, how they are approved, and what patients can expect from treatment. Additionally, these groups actively advocate for policies that prioritize patient interests. Such policies include ensuring that treatment decisions are made collaboratively between patients and healthcare providers, requiring notifications when substitutions are made at the pharmacy, and making sure that any cost savings are passed on to patients. Furthermore, these organizations provide support and community platforms where patients can ask questions, share their experiences, and connect with others. This support can help reduce anxiety related to treatment changes. By empowering patients with credible information and advocating for their interests, these organizations are vital partners in ensuring that the integration of biosimilars into the healthcare system prioritizes patient well-being and confidence. The FDA's regulatory framework for biological products, particularly the abbreviated pathway for biosimilars and interchangeable products, represents a sophisticated synthesis of advanced science and pragmatic public policy. It is a system built upon the fundamental understanding that biologics, due to their origin in living systems, possess an inherent variability that distinguishes them from small molecule drugs. This scientific reality necessitates a shift from a standard of identity to a standard of high similarity, confirmed through a rigorous, stepwise evaluation. The totality of the evidence approach ensures a comprehensive and flexible assessment, anchored by a foundation of highly sensitive comparative analytical studies and supplemented by targeted clinical data to resolve any residual uncertainty. This science-driven process is designed to provide a clear and efficient pathway for follow-on biologics to reach the market without compromising the agency's high standards for safety and effectiveness. The additional requirements for interchangeability, including dedicated switching studies, provide the necessary confidence for pharmacy-level substitution, a key mechanism for increasing patient access and reducing healthcare costs. Beyond the regulatory science, the successful realization of the goals of the BPCI, increased competition, lower costs, and broader patient access, depends on a complex interplay of economic, clinical, and social factors. While biosimilars have already generated tens of billions of dollars in savings, their full potential is constrained by market barriers, including payer policies and patent challenges. The adoption of these medicines relies heavily on the confidence of clinicians, which is being steadily built through accumulating real-world evidence and the clear guidance of professional medical societies. Ultimately, the foundation of this entire ecosystem is patient trust, which can only be secured through transparent communication, robust education led by providers, and the vital work of patient advocacy organizations. Ultimately, the framework, supported by transparent tools like the non-proprietary naming convention and the Purple Book database, is designed to achieve a critical balance, fostering innovation in novel biologics, promoting competition through a robust biosimilar market, and, above all, assuring healthcare professionals and patients that every FDA-approved biological product, whether an innovator, a biosimilar, or an interchangeable, is safe, pure, and potent. This was an episode of the Nexus Clinical Podcast. This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this content is at the user's risk. This is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay seeking medical advice for any medical condition they have, and they should consult their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.